Hi students and welcome to this special discussion on peptide bond formation. So last time we discussed already the 20 standard amino acids, and when we mean standard amino acids, these are amino acids that are usually found in your proteins. So we can also call these 20 standard amino acids as your proteogenic amino acids. So na-discuss na natin last time yung kanilang classification into non-polar, positively charged, polar, and negatively charged. So this is brought by the differences in the side chain. So as you can see, the side chain will dictate the identity of your amino acid as well as the properties of that particular amino acid. But before we will proceed with the discussion on peptide bond formation, allow me to introduce to you our 21st and 22nd proteinogenic amino acid. So we have here selenocysteine and pyrrolysine. So selenocysteine is abbreviated as SEC with one letter symbol of U. So this selenocysteine is an analog of cysteine. So remember in our discussion, cysteine has a CH2SH side chain. But in this case, your selenocysteine, the sulfur is replaced by Se or selenium. So this was found also in humans. So some human proteins to contain this selenocysteine amino acid. So that's why it's called the 21st proteogenic amino acid. On the other hand, we have here pyrrolysine, abbreviated as PYL, or one letter symbol of O. So this can be found in methanogenic archaea and bacteria. So when we say methanogenic archaea and bacteria, these are microbes or microorganisms that give off methane as a byproduct of their metabolism. So you will see here pyrrolysine is similar to your lysine, usual lysine amino acid, but there is this pyroline uh, additional structure. So this pyrrolysine amino acid cannot be found in humans. So in the story, based on my readings, our stop codon is UAG, but in methanogenic archaea and bacteria, this UAG codes for this amino acid. Okay, so this is your 21st and 22nd proteinogenic amino acid selenocysteine and pyrrolysine. So now for the main topic for today. This is all about peptide bond formation. So how do we form this peptide bond formation? This peptide bond formation is actually a condensation reaction. When I say condensation, you are trying to merge two amino acids. So it's a condensation reaction between two amino acids with a consequent release of water. And we call that as your dehydration. So what happens in the formation of your peptide bond? This involves linking of the amino group of one amino acid, the second amino acid here, with the COOH or the carboxyl group of the first amino acid. And in the process, there is a release of water, your dehydration process. So where is now your peptide bond? The peptide bond here is this the bond between your C and N. So this is your peptide bond. So peptide bonds are also called amide bonds. So why? Because in your organic chemistry, you should remember that when you have your C double bond O, N, and N can be bonded to either carbon or hydrogens, this this functional group is called an amide bond. Therefore, in the condensation reaction of amino acids, you are forming an amide bond. And all peptide bonds are called amide bonds. So, so that is the basics of how amino acids polymerize, or how they uh, connect with each other. So let's assume that you have uh, a several amino acid polypeptide. So you have a polypeptide consists of several amino acids. So in this figure, in this slide, you will see here that you have how many amino acids linked together. So you just count, or you just note the number of your functional, your side chains. So we have one, we have two, we have three, four, and then we have five. So that means we have five amino acids linked together, and we will call this your pentapeptide. So remember, you are naming your polygons based on the number of sides. So we will also adapt that uh, naming here. So if you have two amino acids, you will call that your di. If you have three, that's tri. You have four amino acids linked together, that's tetra. Then we have your five, which is penta, hexa, hepta, octa, nona, deca, and so on. So that is how you will classify your polypeptide depending on the number of amino acids linked together. So let's go back to our example here in your pentapeptide. In your pentapeptide, or in any peptide, you should you should know how to locate the ends of your polypeptide sequence. So we have here a term called amino terminal end, and the other one is a carboxyl terminal end. So it is named as such because of the free group. So for the amino terminal end, you have a free amino group here. So this is not used for um, the peptide bond formation. So this is a free amino group. On the other hand, you have a free carboxyl group. So this carboxyl group can also condense with another amino acid, and then the amino acid that was added, that will become now your carboxyl terminal end. So if you're having time, or if you're having a hard time memorizing amino terminal end, you can also use the terms N terminal end. So when say N terminal end, that also refers to the amino group. On the other hand, your carboxyl terminal end can also use the term C terminal end. So C referring to your carboxyl group. Okay? So let's try to identify what are these five amino acids linked together. So this one is CH2OH. So this one is your serine. Serine. This one is your H. So that corresponds to your glycine. This one is, you have the CH2 with the phenyl ring and the OH. So this must be your tyrosine. And then you have your CH3. This is your alanine. And the last one is your leucine. So try as much as possible, try to memorize or familiarize yourself with the structure of the side chain. Okay? So there are rules in naming peptides. So since we have here a peptide, a polypeptide, then we should give a name to this peptide. So first one is to locate the N-terminal residue. So I mentioned that's the free amino group. That's the end wherein you will find the amino acid with free amino group. And by the way, the polymerization, or the formation of your peptide bond, goes from your left to right. So the direction is left to right. That's why I'll just remember that your amino acid should be in L form. So why? Because of the proper orientation of your amino group and your carboxyl group. So imagine if you have a D, so when say D amino acid, your amino group is on the right side of the structure. So if your amino acid is in the D conformation configuration, then it cannot create the peptide bond. So only L amino acid because of the proper orientation of the amino group and the carboxyl group. So going back, uh, you always locate the N-terminal residue for naming your peptide. So after that, you will replace the INE in the amino acid name with the IL for all residues except for the C-terminal residues. So if you have uh, glycine, so let's give an example. You have glycine. This will now become your glycyl. And we will write the whole name of the peptide as one word. So no spaces in between. So whole word. Okay. So before we proceed with the naming, let's go back to the table of the 20 amino acids, standard amino acid. So if you have your glycine, this will now become your glycyl. Your alanine will become alanyl. Your valine will become valyl. Your cysteine will become cysteinyl. Your proline will become prolyl. Lysine will become lysyl. And then arginine will become arginyl. Histidine will become histidinyl or histidyl. We have your phenylalanine becoming phenylalanyl. Your tryptophan will become tryptophyl. Your methionine will become methionine. Isoleucine will become isoleucyl. Leucine will become leucyl. Serine will become serial. Threonine will become threonyl. Tyrosine will become tyrosyl. Your asparagine will have a problem with this. So asparagine will become asparaginyl. So you drop the E, you convert it to YL, asparaginyl. Your glutamine will become glutaminyl. Your aspartic acid and glutamic acid, we will use the terms aspartate, and glutamate. But since we need to change this to your YL, your aspartate here will become aspartyl. Where's my mouse? So that will become aspartyl. And then your glutamate will become glutamyl. So you should be familiar with the name and the corresponding YL name of your amino acids. So now let's apply the rules on the naming of your peptides based on the three rules that we have uh, enlisted or enumerated a while ago. So again, the first thing to do is to locate the N-terminal amino acid. So this one is your N-terminal amino acid. So we already mentioned that this is your serine, your glycine, your tyrosine, your alanine, and then your leucine. So that is the proper sequence of your amino acid sequence or polypeptide sequence. Now, we need to change the name of your amino acids into their YL, except for this one, your leucine. So your leucine here will stay the same. So your serine will become serial, your glycine will become glycyl, your tyrosine will become tyrosyl, then alanine will become alanyl, and then leucine will stay the same. So there's a rule that the name should be written as a whole word. So it will become serial, glycyl, tyrosyl, alanyl, leucine. So that is how you will name your polypeptide using the YL names. So you change the name, you change the INE, the in of the amino acid to its ill name, especially if it's not the N, rather the C terminal amino acid. Now, there's also another way of naming your polypeptide, your pentapeptide shown here. So imagine if you have a uh, very long amino acid sequence, let's say you have 100, then you will write everything in this format, your ills. So that is very cumbersome or very matabaho siya. So that there's another way to make your naming simpler. So this time we will use the three-letter abbreviation of your amino acid. For serine, so that will be ser. Then you have your glycine, gly. Tyrosine is tyr, tyre. And then alanine is ala. And then leucine is leu. So now what is the convention in naming your pentapeptide using the three-letter abbreviation? So you will place or you will put a dash in between amino acid residues. So it will become like this. So you have your ser, gly, tyre, ala, leu. So if you encountered an amino acid sequence or a polypeptide sequence, this ser is the first one. So this one is your N-terminal side, and then your leu here is your C-terminal side. So what if you have around 500 amino acids in a sequence? So writing the name as it is with the ill ending and the three-letter symbol is also still not the good um, way or approach to name your amino acid. So medyo matabaho yon. So we can simplify your amino acid sequence by using this time the one-letter symbol. So we will give the symbols of the symbol, the one-letter symbol of this amino acid. So your serine is S, your glycine is G, your tyrosine is Y, your alanine is A, and then leucine is L. Are given a symbol of L. So that means your amino acid sequence will be SGYL. So again, if this is your if this appears first, then this is your N terminal side, and then or N terminal amino acid, and then your L here is your C terminal amino acid. Okay, so the point here is you should know how to name your amino acid sequence, your polypeptide sequence, based on three things. So the individual names of your amino acid, the usual name, then you change it to ill. Then you should also be familiar with the three-letter abbreviation of your amino acids and also the one-letter symbol. So you should, all in all, dapat alam nyo kung paano i-drawing or familiar kayo with the side chain, uh, with the structure of the side chain of each amino acid, its name, its three-letter abbreviation, and the one-letter symbol. Okay, so that ends our short discussion on peptide bond formation. So thank you very much.